DNA. Those are 37 genes that already stay here inside the mitochondria. And then some genes transfer to the nucleus. Those genes are called nuclear encoded mitochondria genes or NMT genes. Those genes actually stay in a single copy or got duplicated, so we have another copy of those genes inside the nucleus. The genes are transcribed inside the nucleus, translate inside the cytoplasm, and then move the protein imported to the mitochondria to interact with the mitochondria proteins. The interaction between these two organelles is not only important for energy production, but also is important for replication, translation, transcription, transport, and communication. Actually, we have 4 levels of the interaction between the NMT genes and mitochondrial genes. We have the protein-protein interaction happening inside the octopus complexes. We have 2 other types of the interaction, the protein with the RNA genes, with the mRNA, and binding factors that happening in other layers of this interaction. In 2010, our lab studied the NMT genes in Drosophila melanogaster. And then Gayok, the postdoc in the lab, found we have 23% of the gene clustered into the gene families in Drosophila melanogaster. Those genes got duplicated either by the DNA or RNA-mediated duplication events. And regardless of the reason of the duplication or the method of the duplication, they have high energy-related functions. They are super old, the genes are super old, as they are present in all Drosophila species. And then when we look at the expression of these genes, we realize that, or they realize that 54% of these duplicated genes express highly in tissue-biased manner. And then all of those tissue-biased genes are testis-biased genes. So if I show you a heat map of these gene families, sitting here on the y-axis and then on the y, on the x, you can see the tissues in the adult flies. The darker the color, the higher the expression. So you can see we have a lot of dark colors right here. I can zoom in one of the families. Most of the time, families have 2 members. The parental gene, which expresses highly in every tissue, showing here in red on top. And then the duplicated gene only highly expressed in the specific tissue here, testes, which is true for all of these genes. And then you can see the pattern, they are highly expressed in testes. Also, these duplicated genes are expressed high in the meiosis, while the parental gene of this duplicated expressed earlier in the germline. Some of these genes have been found to be under positive selection, and then they are mostly present in Drosophila sperm proteome DSP. But the question another question in the introduction is how these genes are expected to evolve. Because we have the interaction between the mitochondria and the nucleus, and the mitochondria imported most of its protein from the nucleus or from the NMT genes, the interaction between these 2 organelles is crucial for the function of the mitochondria. However, these 2 organelles evolve differently. Mitochondria evolve faster than the nucleus, and the mutation rate in mtDNA is higher than the nuclear DNA. So there should be a way to maintain this crosstalk between mitochondria and nucleus. So there are some possible ways to maintain this crosstalk. Maybe they co-evolve with the mitochondria. The NMT genes in general co-evolve with the mitochondria, which has a higher mutation rate. So we expect to see a higher mutation, a higher evolution rate compared to the other genes. They might be there to compensate for deleterious mitochondrial mutation, so response to that. And then they might be there to respond to male harming mutation because the mitochondria is inherited maternally, so only the females can select the mitochondria. Actually, if you look at the slide, in the population, in the mitochondria population, we have 3 different alleles. We have the neutral allele, we have the female harming allele, and then we have the male harming allele. And then only the female harming allele cannot pass the selection. The rest of the alleles can pass the selection and go to the next generation. That's the reason we call it feminized mitochondria. Females don't care about the male. We might have received a negative fitness effect allele in the males, which can have a negative impact in the males. So, 3 different hypotheses have been proposed for these duplication genes. These duplicated genes might be there to just cope with male harming mutation. This is known as the Roger's hypothesis. Another hypothesis is that these genes might be there to, these genes have a new function or a specialized function, here sperm function or male function, to have a kind of better sperm or more energetic sperm for the males. This is known as the Guilloc hypothesis. And then simply, maybe those genes are there because we just need more of the same protein. So I'm going to talk about four different stories. As I told you, the stories are all over this set of the genes, NFT duplicated with testis bias expression genes. And then we were wondering how common is this phenotype? How often we can find the NMT with the testis bias expression in other species? Also, we wanted to see how these duplications are evolving in the population, what function they have, and then because as they have energy-related function, often it comes with the higher ROS production. So we wanted to see if they are different in the ROS production level. So, to answer to the first question, we studied the NMT duplication in human, and then we expected to see the general pattern in the human like any other species, the same as we saw in Drosophila. We expected to see a big number of duplication with testis bias expression. And we picked the human genome because it was the best annotated genome available, and then it has the expression data available also. So we followed this pipeline, we created gene families, and we assigned the expression and the age of the genes. We did the same for Drosophila melanogaster to compare the results of the human to Drosophila melanogaster. And here is the table that's showing the human data on the left. You can see the human data, they have a lower number of the duplication compared to duplication in the whole genome, which is different than Drosophila melanogaster. The genes are older in both species, the NMT duplication genes, and then they relocate more often in both species compared to the other duplication in the genome. However, the big difference between these two was about the expression. If you look at this heat map, you can see on the left the Drosophila melanogaster heat map expression. So you can see a lot of, so on the Y, you can see the duplicated genes only, and then on the X, you can see the tissues. So you can see a lot of duplicated genes highly expressed in testes, while in human, only we have a couple of testis-specific genes, and testis was not the only tissue has the bias expression. We saw liver, skeletal muscle, and other tissue specificity among the human NMT duplicates. Next, in the next step, we wanted to test the male harming mutation hypothesis or Roger's hypothesis. So the best place to test this hypothesis is in the Oxford complexes, when the proteins encoded by the two different genomes interact directly to each other. So we counted how many of the testis bias duplicated genes are interacting with the empty proteins or the other way. And then we couldn't find any support for that hypothesis, neither in human data nor in Drosophila melanogaster. And then we look after the reasons for these differences between the species. As you may know, we have the male-male competition in the Drosophila, which is missing, kind of missing in the human. So we are thinking that the flies have a stronger selective pressure compared to the Drosophila melanogaster. Also, the Drosophila melanogaster have bigger effective population size compared to the human. When we look at the distribution of the mitochondria in the sperm, the two species are different. in the human, we have the mitochondria only in the neck or the middle piece of the sperm, while in the Drosophila, the mitochondria are all across the tail of the sperm, showing here in green. So you can see the distribution is different. And then lastly, they use the mitochondria differently for sperm mobility. The Drosophila melanogaster relies on the oxalose complexes energy, but the mammalian and human relies on glycolysis and oxalose complexes for sperm mobility. So to summarize this story, we have fewer, younger, with different function, NMT duplicated genes in human compared to Drosophila melanogaster. The testes are not the only tissue that shows the bias expression. We saw the bias expression in other tissues. And then in both species, the duplicated genes often has the housekeeping as a parental gene. And then we couldn't find any support for the male harming mutation compensation hypothesis or Rigel hypothesis in this study. The next story, we were interested to see how the testis bias duplicated genes are evolving. The duplicated genes are quite divergent from the parental. They are only 62% identical in amino acid level. And then when we look at the Ka-Ks ratio for the testis bias duplicated genes, we found they are actually higher than the parental. So they evolve faster than the parental genes. And then we wonder maybe that high ratio is because of the expression. So we compared the Ka-Ks for the NMT genes to the random set of the testis bias gene. And then we found that the faster evolution of the genes is explained by their expression. So we couldn't find any support for the male harming mutation here again, because if the male harming mutation is true, we expected to see higher. We expected to see a higher ratio in the NMT testis-specific genes compared to the testis bias genes. Using all available polymorphism data in Drosophila melanogaster, simulants, and Yakuba, and performing the McDonald treatment test, we were able to find some other genes that under positive selection in this set. And then they found some of these duplicated genes have extra mitochondrial localization signal that the cleavage site is proposed to be important for protein stability. And then we are thinking that's another reason they are different than the parental. Next, we look at the ERC value, evolutionary rate correlation between NMT genes and the parental, and we found that the NMT genes co-evolving with each other higher than the parental genes. And then because we expected all of those genes have the mitochondrial function, we couldn't find any strongly coevolution between different classes of these genes. By that, I meant the single copy, duplicated genes, and the parental genes. So we didn't find any support for the coevolution of those genes with each other. The next question we had was if there is any coevolution between the NMT genes and mitochondrial genes. The best way to answer that is just calculating the linkage disequilibrium between the genes. But we found that the recent invasion of Wolbachia in the Drosophila population might swipe away the mitochondrial variation. The Wolbachia is a bacteria that can infect the Drosophila and many other insects. And then the female that has the infection has higher fitness effect compared to uninfected female. So that mitochondria can spread more often than the other genes. So we try to study the differentiation between NMT genes in the Drosophila between the populations, and we use the FST here for this analysis, looking at the FST value for the retrogenes in 9 Drosophila populations. And then we didn't find any significant differences between the testis bias NMT genes and the random set of the genes. And then this means that we don't have any support for the higher or lower differentiation between populations. So to summarize this study or this story, the NMT testis bias duplicates evolve faster than the parental, and they are often under positive selection. We show sign of specialization because they have additional protein cleavage site. And then they don't show any sign of the integration or adaptation in the reasonable backyard invasion because of the reasonable backyard invasion. We expected to see that either the adaptation or integration because after the invasion they might co-integrate with the NMT genes, or NMT genes might respond to that change. But we didn't find any support for neither of those genes. And then all of these proposing, suggesting that the NMT genes might have been selected for different functions in the male germline. So the third story is actually about the functional study of these genes. And no doubt we used the Drosophila melanogaster, the Cinderella of the genetics, for this study. We expected to see the male infertility in the knockdowns and knockouts. For most methods. And then we expected to see lethality when we ectopically express these genes in soma. So to study the function of these genes, we took three different approaches. We have the RNAi knockdowns, we have the CRISPR knockout, and we also drive the expression of the gene to study the function of this set of the genes. So the first approach was the knockdown using US GAL4 system. GAL4 is the yeast transcriptional activator that can be used in a tissue-specific manner, so you can drive it in your tissue of interest only. When you put the GAL4 beside the US in the same background, it's going to produce a hairpin RNA. The hairpin RNA is going to process to the short interference RNA. And you're going to go and find the mRNA of gene of interest, and then degrade that mRNA. So you're going to have the decrease in the expression of the gene of interest. For this study, we have received the lines from the BDRC. We actually have two different libraries in the flies community from the BDRC, the GD and KK libraries. They are different in the insertion site. Also, they are different because they are targeting different regions of the gene of interest. And then if they show the same result, meaning that we can use those data better than only one library. So we have 68 lines for 39 genes. Those are all NMT duplicated with testis bias expression. And we use the BAM-GAL4 as the germline driver for this study. Reciprocal process at two different temperature was performed. The second approach was the knockout in vivo with the treat cow flies. So here in these flies, they have the GAL4 Cas9 in the background. When you cross it with the collection of the treat cow, the Cas9 gonna bring the single guide RNA to the gene of the interest. And it can make the cleavage, which can get repaired by the non-homologous enjoining, potentially creating insertion, deletion, mutation. This method is super efficient, and each time produces a new mutant. We received the lines as a gift from the Harvard Medical School, and we used two different drivers to drive these genes. The actin 5C GAL4 Cas9 to drive in soma and germline, and then the nanos just for the male and female germline. And only we have these two cow lines for 7 genes. Also, we use the mitophage web server to find the localization signal, the mitochondrial localization signal of these proteins or genes. Simply this website looking at trying to find the putative mitochondrial pre-sequences and the cleavage sites, and it can give you the probability to import into mitochondria or localize it inside the mitochondria. So when you look at the distribution of the NMT genes or NMT duplicated genes across the mitochondria compartment, you can see the duplications are in the oxosamplexes, outer and inner membrane, in the carriers, the TCA cycle, the redox activity, and also in other activities. And you don't see the duplication in transcription or translation machinery here. So I'm going to show you the results of the knockdown and knockout based on different compartments. The first is the oxphos complexes. The proteins in these oxphos complexes encoded by the mitochondria and the nucleus. So we have an interaction between these two proteins, encoded proteins from two different genomes. We have a GD and KDK library for some of the genes, not for all. And then we had 12 genes here for this set. And the fertility effect, the fertility test with the PAM, didn't find any fertility effect for the complex 1 and complex 2 genes, but for complex 3, 4, and 5, we see different degree of the male infertility. And then you can see the probability of localizing to the mitochondria. They are pretty high because they are importing to the mitochondria. And then whenever it was possible, we dissected the testis of the knockdown that shows the infertility. And on top, you can see the control, the normal testes. This is the tip of a testis. They have the germline over here. And then they are getting mature. And then we have the sperms in the bundle. They get individualized. And then they deposit to the seminal vesicle. We saw two different phenotypes in the knockdown. For the first phenotype, we didn't see a sperms in the bundle. And then for the second phenotype, we saw the sperms in the bundle, but we didn't, the seminal vesicle was empty, actually showing here in this picture. So on the top panel, we can see the full seminal vesicle in the control, and the sperms are moving. We have the mature, complete individual sperm here, but we are missing that in this below panel. So this is the knockdown. The seminal vesicle is empty, and the sperms are piling up here before the seminal vesicle, suggesting that the individualization step is interrupted in those knockdowns. Looking at the membrane of NMT duplicated genes in the membrane, we saw some male infertility and some without any fertility effect. Usually those genes have lower mitochondrial localization probability because they are not importing to the matrix of the mitochondria. They are staying in the membrane. The TCA cycle was the next compartment. We found the semi-male sterility for 2 genes, not for the other 3 genes. And then the redox compartment, we didn't see effect for many of those genes. We just saw the male sterility with the defect in the individualization stage in these plots. And then the last compartment was the other activities. Some of them showed male sterility, while others didn't show any effect. So to summarize the RNAi results, 41% of the knockdown shows the male sterility. The trip cow lines always confirm the RNAi fertility results. And then when we look at the literature, we have like 6 null mutant for some of these genes. So some of these genes have been mutated before, and all of those shows male sterility consistent with our RNAi results. In the next, we decided to look to see if we can find any association between the presence of these genes in the Drosophila sperm proteome and fertility results. Meaning that if the gene is in the Drosophila sperm proteome, it shows the male sterility. We couldn't find any support for that association. And then when we are knocking down a gene of the interest, the parental gene is still expressing in that mutant or in that knockdown. So we were wondering if the parental gene is compensating for the duplicated genes. So we simply counted how many of the parental genes are in the Drosophila sperm proteome, and then they don't show any effect. and we couldn't find any association again between the fertility effect and the presence of the parental indrosophila sperm protein. However, we saw unexpected lethality effect in 36% of the RNAi knockdown. When we look at the expression of these genes, so we drive these genes also with actin 5C in soma and germline, and that's where we saw the lethality effect. When we look at the expression of these genes, they are moderately to high expressed in larva stage. So we try to find if there is any association between the larva stage expression and the lethality. And actually we find if they are higher in larva stage expression, if they have higher expression in larva stage, the chance of being lethal is higher. But the lethality wasn't confirmed by 3 cow lines for 7 genes. And then this is still puzzling to us why they show the lethality. Some of those knockdowns of having off target, meaning that they are targeting other genes, so that can explain the lethality. And then when you are driving the RNAi, you are stressing the cell that can cause the lethality. So this is two explanation for the lethality, but we need to create a new mutant to confirm the real function of these genes. So at this point, we cannot say we are not sure about the function of these genes. The third approach in the functional study was ectopically express these genes in soma or overexpress these genes in germline. We expected the ectopically expression of these genes be lethal because it produces more ROS, or it produces something that is not good for the cell, so it could be a lethal phenotype. Taking advantage of the polymorph and also the 3 OE lines, we were able to ectopically express empty duplicates in soma with 2 different galford drivers. And then when we ectopically express those genes, they were lethal for these 4 genes. However, when we overexpress these genes in the germline, they were not lethal. So we didn't see any effect in the overexpression of these genes in the germline, but it was lethal in the soma, suggesting that they have different function in both in the soma and germline. So to summarize this story, the knockdown and the knockouts often need to be made in fertility as we expected. We saw either the individualization stage defect or allele defect in those knockdowns. And then the parental gene is not compensating for the duplicated genes in the fertility knockdowns. And then the ectopic expression of these genes is lethal in soma, while it's not, it's without any effect in the germline. So the last story that I'm going to talk about today is about a specific gene called COX4L. The COX4L is a duplicated gene from the COX4. They are 52% identical in the protein level. And then the COX4L is found with the Drosophila sperm proton, while the COX4 was not there. When we look at the expression of these 2 genes, we saw the COX4 is highly expressed in nearly every tissue, while the COX4L is highly expressed in testes. So we look after the COX4L and try to find the COX4L, the duplicated gene, in the other species. We found the COX4L in all the Drosophila species and some close related species, but not for the others. And then the COX4 was present for all Diptera species. And here you can see these 2 genes clustered separately because they are missing in some species, but are presenting in all of the species. And then the COX4L is evolving faster. That's another reason it's clustered differently than the COX4. And then when we look at the present or the absence of these duplicated genes in the Diptera we suggested that the Cox-Form duplication node is in this, which brings the age of this ore to 245 million years. Then we were interested to see if the COX-4 is actually localizing inside the mitochondria. We didn't have any data, so we derived the COX-4L fourth line with the BAM-GAL4 driver. Those fourth line has an HA tag, so we can stain the HA and see the localization. In this panel, the testes were dissected and stained with the DAPI for the nucleus. For the mitochondria, red was used for the mitochondria, and this is the green, the HA tag, and that's the overlay of all of those colors. The yellows in this picture showing that the gene is inside the mitochondria. So we can confirm the COX-4 protein is localizing inside the mitochondria. Then we wanted to study the function of this gene by the knockdown. So we used the GD and KD library for these genes. Those libraries didn't have any off-target. And then we performed the viability test and fertility test. For the viability test in the knockdown, we didn't see any effect compared the knockdown compared to the control. The blues are the male, the red is for the female, and this is the GD library and KD library. And then for the fertility test, however, the males was semi-sterile or infertile in both libraries, while female are doing better compared to the control. So we decided to go further and create a knockout of this gene. So we designed two different guide RNA that can target the two sides of the gene, remove the gene, and replace the gene with the DS red. So this is the CRISPR-Cas9. And then when the company injected the G plasmid inside the posterior position of the embryo, we screened the flies and looked after the flies with the glowing eye, meaning that the gene has been replaced with the DS red. So we performed the viability test in the knockouts, and we didn't see any effect in the males and females of the knockout compared to the control. But when we performed the fertility test, we saw the COX-4 males are completely infertile. And then the females still doing better than the controls in the knockout. We dissected the testes of those knockout flies, and then we confirmed that the defect is in the individualization step. On this panel, on top panel, you can see a control with the full seminal vesicle. And those are individualized sperms moving around, but we are missing that the seminal vesicle is empty, and then we have the sperms in bundle inside the knockout flies, suggesting that there is a defect in the individualization stage of spermatogenesis. Next, we decided to rescue this phenotype to make sure this is the real effect of the gene. We were able to completely rescue the male infertility with the BAM and nanos GAL4 drivers. And the females didn't change when we rescued, we performed that analysis for the females. In the next step, we overexpress or ectopically express this gene in the soma and germline. When we ectopically express this gene in the soma, it causes mortality, so no flies at all. But for when we, and also when we ectopically express that gene in the female germline, we didn't see any effect. But when we overexpress gene in the male germline, we saw a reduction in the male fertility, which suggested that the fine-tuned expression of the COX-4L is necessary for the male fertility. We went a little bit further to dissect the lethality effect. So we wanted to see in which state the lethality is happening. We didn't see the larvae of the pupa arrest, so we decided to collect the larvae of the control and the overexpression flies. We didn't see any effect over there. However, when we collect the embryos of the control and the overexpression flies, we saw a reduction in the overexpression, suggesting that the lethality is happening early in embryonic stage. These of the flies in the knockout, we were wondering how much ROS protein the testes in the knockout does have. So we stain the testes with the vacuum for the nucleus, with the mitotracheal red ROS in red for mitochondria, and then we have an overlay picture here. The mitotracheal red ROS can measure the ROS production of the mitochondria. So the higher the red color, the higher the ROS production. And then you can see in the COX4L on the top panel, we have more red on this mitotracheal compared to the control, suggesting that we have an increase in the ROS production in the knockout of COX4L. Furthermore, we wanted to see the functionality of the mitochondria. So we stained the mitochondria with the TMRE, which measure the membrane potential of the mitochondria. And then here we have the COX4 knockout on the W on the bottom. And then you can see on the bottom we have a lot of red colors, meaning that the mitochondria is functional. However, this is completely missing on the knockout, suggesting that the mitochondria is not functional in the knockout. This data was also confirmed using the mitotracheal deep red, which can measure the membrane potential of the mitochondria. So we are thinking in the normal germline mitochondria, we have all of those complexes, and the electron is transferring between the complexes to generate the ATP. However, in the knockout germline mitochondria, we are missing the COX4L, this part of this complex 4, which leads to the leakage of the electron, which eventually can cause an increase in the ROS production. An increase in the ROS production can change the membrane potential of the mitochondria and then eventually cause non-functional mitochondria. So to summarize this story, the COX4 is a well-conserved sperm protein differentiated from the COX4L based on the current data. We are planning to rescue the phenotype of the COX4L with the COX4, with the parental gene, to make sure they are really different. But this is one of the things that left. I'll just show you the COX4L protein localized inside the mitochondria. This is consistent with the Drosophila sperm proteome data. The COX4L is essential for the spermatogenesis and male fertility, as the COX4L knockout is completely infertile. And then we are thinking an increase in the ROS production can lead to the increased change in the membrane potential, and then eventually the mitochondria are going to lose the functionality, which can perturb the individualization step of spermatogenesis and cause male infertility. So to wrap up the whole presentation, I have answers for the questions that I asked in the beginning. For how common those genes are, we couldn't find those genes in other species, suggesting that Drosophila melanogaster use those genes in a specific or specialized way. The distribution of the mitochondria in the tail of the sperm might be one of the explanations for that phenotype. For the evolution part of, for the second story, the evolution, the genes are evolving faster and often under positive selection. And then for the function of those genes, we are thinking those genes are important for the male germline, and they have a specific function only in the male germline, not any other type of the cell. And then for the ROS production, we couldn't test that hypothesis because the ectopic expression of those genes is lethal in the soil. So we wanted to ectopically express those genes in the skeletal muscle or other tissues and measure the ROS production, but that lethality suggests that we cannot do this kind of analysis. So with that, I want to acknowledge my advisor to give me the freedom to test every single crazy idea I have during these 5 years. Also, thank Chabi for running our lab, our former members, Anya and Divash, and then Aiga, Fatima and Mira, the current students. Also, the Victor helping me with the creating the knockout from the British Southwestern. I have an army of the undergrad students helping me with this project, so thank to them. Also, thank to my committee to support me for this. And then I should thank Mark for providing the reagents for the staining of the mitochondria and then helping me with the staining, his lab and himself. And then I should thank the funding. This project was initially funded by NIH, but the Phi Sigma money helped me to perform the COX-4 knockout and staining, so it has a big impact in my research. Also, I used a lot of resources in this presentation, thank to all of them, and then thank to the collaboration for this. And last but not least, I want to thank my wife, Anas, and my little daughter, Ariana, for supporting me to start, continue, and finish my PhD. And with that, I take your question. Continuing these studies, one of the things, as I said, left is knocking out the COX-4, and then also that's one of the projects we also trying is not offline for the and generate the offline for the COX-4, so we can test if the parental gene is rescuing the duplicated gene function. I would like to do the knockout, to have a knockout of some of those RNAi with the lethality, but we can, I think we can continue that today.
So you could add more?
Yeah, I'm a chemist, so I understood a little bit of what you were saying. And, when you got to the bar graphs, I got really excited. So the error bars and the bar graphs, how do we get them determined?
So those are error bars. And then we have the replication for all of those tests, at least 3, sometimes 5. So for every single temperature and every single cross.
The question, like, if I don't care about snakes or lizards, what does your dissertation tell me? And so you didn't find this pattern specifically.
So still, I am thinking this is a pattern in Drosophila melanogaster, in Drosophila species. So the mitochondria compared to the number.
Families more or
they are duplicated, but they are used in the industry. So if you match it, also can explain that those duplications are there to provide more energy for the sperms to swim faster to get to the egg and then be successful. So those are other explanations for that. But these genes also are under positive selection. But we don't have.
So that all of the data cascade vaccines, that's pretty strong. The slight differences in the degree.
Yeah, I have the data. I think it's in the supplementary table chapter 4 in the dissertation.
I'm asking because I'm kind of wondering if the MK test showed stronger.
Yeah, we have at the end